Good evening, everyone. Welcome to the captivating 11th edition of the online lecture series proudly hosted by the esteemed Physiological Society of India, launched on July 13, 2023. This series is designed to complement the curriculum of physiology courses at both undergraduate and postgraduate levels. Organized by the Educational Committee of the Physiological Society of India, these lectures are scheduled on the fourth Saturday of each month during the evening session from 7 to 9 p.m. Tonight's lecture, the 11th in the series, delves into the intricate workings of the cellular physiology. Today's enthralling topic will be illuminated by two luminaries in the field. It gives me immense pleasure to announce the esteemed presence of our distinguished speakers, who are undoubtedly the epitome of knowledge and expertise. With bated breath, we await the insightful words from Professor Liayum Amarnatham Sharma and Professor Govindaraj Yangkhom. In the first session, we have the privilege of delving into the realm of the cellular physiology under the erudite guidance of Professor L. Amarnath Sharma. Dr. L. Amarnath Sharma, a distinguished physiologist, he earned his MBBS from Manipur University in 2002, followed by an MD in physiology from the same university in 2018. Additionally, he completed a basic course in biomedical research through NPTEL and a basic course in medical education in November 2023. Professionally, Dr. Sharma has served in several notable capacities. He was a senior resident in the Department of Physiology at the Regional Institute of Medical Sciences, Imphal, from November 2020 to March 2022. He has been an assistant professor in the Department of Physiology at the Tomo Riba Institute of Health and Medical Sciences, Nahar, Naharlagun, since March 2022. Additionally, he has served as a senior medical officer in Manipur Health Services since January 2011. Dr. Sharma has actively contributed to medical education and research, and his research publications include studies on iron deficiency anemia and anemia among adolescent school children in Manipur, and the hormonal profile of women in different age groups. Through his extensive education, professional experience, and research contributions, Dr. Liayum Amarnath Sharma has established himself as a dedicated and accomplished figure in the field of physiology. Sir, you may now commence your session. The platform is yours to lead. Thank you,
Thank
sir.
you very much,
Please
ma'am.
start your session.
Okay. Good evening, dear students. Today, I'm delighted to give a presentation lecture on the topic Overview of Cellular Physiology in relation to medical physiology. Now, to start with, let me just begin with the next slide. Next slide, please. Now, coming to a brief introduction of the cell that you have learned in your earlier classes also, I just want to give a brief introduction to all of you. Uh, Cells are basically the fundamental living structural and functional unit of all organisms, including human beings. Now, in humans, of course, the cells are highly specialized in both structure and functions because they need to fulfill unique roles and contribute towards varied functional capabilities of the human organism. Different types of cells that we have include For example, the neurons, the epithelial cells, uh, the endothelial cells, so on and so forth. So there are about 200 such specialized cells that basically make up our human body. Next slide, please. Next slide, please. And these 200 different types of specialized cells, they carry out a multitude of functions that basically help each system of our human body to meet the requirements to function as a human organism. However, these cells, even though they carry out different functions, they share certain key structures and functions that support their intense activity. Now, let us now try to understand the different components of a cell, their functions, and its relevance in medical physiology. A cell consists of basically three fundamental components: the cell membrane, the cytoplasm, and the nucleus. As you can see in this diagram, it is a diagram showing the electron microscopy of a cell, eukaryotic cell. Now here you can see this portion, which is the envelope of the cell, is known as the cell membrane. Now inside the cell membrane, all these structures are there. In the structures that are present inside the cells basically include the nucleus. Of course, it is occupying almost a central position in this diagram. And between the nuclear envelope, which is the nuclear membrane that covers the nucleus, and the plasma membrane, we have got a gel-like material that is known as the cytoplasm. Now, the cytoplasm basically consists of water mainly. Around 80 to 90% of the cytoplasm is made up of water. And in this water medium, many solutes, soluble solutes are suspended, enzymes are suspended, and then little structures known as cell organelles are also suspended in this cytoplasm. Now, coming to, please, the next slide, please. Now, coming to the first component of the cell membrane that I have just mentioned, that is the cell membrane. Cell membrane is basically the membrane covering the cell. Uh, in this diagram of a cell membrane that is being depicted, it is known as the fluid mosaic model of the plasma membrane. This was postulated by Singer and Nicholson, and this is the most accepted model of this cell membrane so far. Now, according to this model, what we see in this picture is that the cell membrane consists of basically a lipid bilayer structure, lipid bilayer. Now, in this lipid bilayer, we have got many proteins, glycoproteins, glycolipids, and cholesterol. Now, the lipid bilayer is arranged in such a way that the polar head of the lipid, basically they occupy the outer aspect and the inner aspect of the cell. That is, they will be facing towards the extracellular fluid or the intracellular fluid. Now, that portion of the lipid bilayer is known as the head, and of course, the tail portion of the lipid bilayer, they are basically facing towards the interior of the cell membrane. Now, these tail portion are non-polar, meaning they don't have any pole charge, so to say. Now, in this lipid bilayer, as you can see, there are many proteins. Basically, two different types of proteins are present here. One is known as the integral protein, and the other one is known as the peripheral protein. Now, integral proteins are basically will be occupying the interior of the cell, maybe on one leaflet, and then they may traverse through the cell membrane also, just as you can see here. These are certain integral proteins that are shown here, as I am showing with the cursor. And of course, these are also integral proteins, and these are the peripheral proteins that we can see. Some examples of integral proteins would include the carrier proteins, the ion channel proteins, and then you have the transport proteins, and so on and so forth. And the peripheral proteins, some example will be enzymes, and of course, some may act as receptors too. So the detailed function and structure of the cell membrane will be discussed by the next presenter in the next lecture. So I will not go into the detail of the cell membrane right now. So moving to the next slide, the cytoplasm that I have just mentioned about. This I have already told. It is a gel-like material between the cell membrane and the nuclear membrane, and comprises basically of water, around 80 to 90%, dissolved solutes, suspended particles, and of course the cytoskeletal structures that will include the microfilaments, intermediate filaments, and the microtubules, and then the little suspended structures that we call as organelles. Now, moving on to the next slide, the third component of the cell, that is the nucleus, it is the largest double membrane bound extracytoplasmic cell organelle. Many a time students get confused whether the nucleus is an organelle or not. Now, I want to basically make all of you clear that the nucleus is also a cell organelle, but again, it is considered to be an extra cytoplasmic cell organelle. So, rest of the other organelles, cell organelles, are cytoplasmic cell organelles, whereas the nucleus is an extra cytoplasmic cell organelle. Now, the nucleus basically contains the genetic material, genetic information that is required by the cell for varied functions in the form of chromatins. Now, these functions that the genetic information basically does is controlling the metabolism of the cell, cell growth, and of course, differentiation and division of the cell. Now, these chromatins, when they are highly condensed and organized, they form what is known as the chromosomes. Now, chromosomes are usually not visible when the cell is not dividing. What we see is the chromatin material, okay, when the cell is not dividing. But when the cell is about to divide, these chromatins will organize themselves in such a way that they are visible as chromosomes. So these things I think should be clear to all of you. Now, chromosomes, between the difference between the chromatins and the chromosomes. Now, in human beings, we have got 23 pairs of chromosomes in all the somatic cells. But in case of germ cells, that is the ova and the spermatozoa, they are not diploid; they are haploid. So that is an exception. Okay, coming to the chromosomes are basically giant single molecule of DNA containing thousands of genes. Now, these genes, which are basically going to form the DNA molecule, they correspond to translation of a protein, particular protein that controls cellular structure and function. Now, the second component of the cell nucleus is the nucleolus. Now, as you can see in this diagram, this is a nucleus, and then it's an electron micrograph of a nucleus of a cell. Now, in this, what you see is a condensed condensed structure within the nucleus. This is known as the nucleolus. So, it is a patchwork of granules rich in RNA. Now, the nucleoli, they are more prominently visible and more numerous in growing cells compared to other non-growing cells. The nucleolus basically functions as the site for ribosome synthesis. Now, coming to the nuclear envelope, that is the membrane that covers the nucleus. Nucleus is a membrane-bound organelle. Okay, so the membrane that covers the nucleus is known as the nuclear envelope, and just like the cell membrane, it is also a double membrane structure. Double membrane structure. Okay. However, in between this double membrane structure, there are gaps, so to call as nuclear pores, through which proteins and messenger RNA can travel, okay, from the nucleus towards cytoplasm. Many proteins also enter the nucleus from the cytoplasm via these nuclear pores. Okay. Coming to the next slide. Now, let me just tell you in brief, because I'm covering an overview of the cellular physiology. So, I'll just not go into detail of everything, but let me just start with the cell organelles, cytoplasmic cell organelles, one by one. So, coming to the first cytoplasmic cell organelle, that is the mitochondria. Okay, mitochondria, it is a sausage-shaped, cytoplasmic organelle that is found scattered all around the cytosol. Now, when I say sausage shape, many new research uh, uh, publications have mentioned that the shape of the mitochondria can be dynamic. Dynamic in the sense that it may be changing its shape from the sausage shape to some other shapes too. The most important shapes that is talked about in the journals at this point of time are the punctate shape and the reticular shapes. The punctate and reticular shapes, their interconversion is nowadays thought to be essential for proper functioning of the cell. So that is what I mean by dynamic. Now, the mitochondria, they are higher in number in those cells that utilize ATP at a high rate. So, say for example, muscle cells, when they are exercising, okay, they require larger amount of ATP. So, muscle cells will definitely have higher number of mitochondria compared to other cells which do not utilize that much amount of ATP. Next, they also have the ability to self-replicate. That is one characteristic feature of the mitochondria, that they can replicate by themselves during increased cellular demand or before the cell division. Now, this is something that other organelles, cytoplasmic organelles, cannot do, but the mitochondria does. Besides this, the second characteristic feature of the mitochondria is it exhibits maternal inheritance. So, mitochondria are basically inherited from the maternal side. Other organelles, cytoplasmic organelles, they are inherited both from the uh, maternal as well as the paternal side. Going to the next slide, coming to the structure of the mitochondria, the mitochondria consist of an outer membrane, as you can see in this picture, an outer membrane, and then of course an inner membrane. Now, inner membrane, between the inner membrane and the outer membrane, we have the intermembrane space. Now, the inner membrane, as you can see in the diagram, has got infoldings. Okay, so many infoldings you can see in this diagram also. Now, these infoldings of the inner membrane, they are known as cristae. Okay, now inside the inner membrane, what you see is the central matrix space. Now, enzyme complexes that are responsible for oxidative phosphorylation, that is basically the physiological process by which ATPs are generated. Okay, the oxidative phosphorylation, the enzymes responsible for that, they are lined up in the cristae, and these infoldings provide an enormous surface area for the chemical reactions that are going to be involved in aerobic phase of the cellular respiration. That is why we have these infoldings in the form of cristae in mitochondria. Okay. Now, mitochondria are integral in metabolism of carbohydrates, fats, and amino acids, in that they house all of the components of oxidative phosphorylation, Krebs cycle, urea cycle, fatty acid oxidation, key proteins of ketogenesis, and of course, they also do the function of triacylglycerol synthesis and gluconeogenesis. Mitochondrial, please let us go to the next slide. Now, coming to that was the first role, the function, first function, that is generation of ATP through aerobic respiration. So that involves all this uh, biochemical processes that I have just mentioned. Now, coming to the second function of the mitochondria, that is the role in apoptosis. No, sir, let the first slide be there. Now, regarding the role in the regulation of apoptosis, mitochondria has a very big role. Have you heard of apoptosis before? I think it is a new term to most of you. But apoptosis, let me just say in simple term, it is basically the process of cell suicide, or also known as the programmed cell death. Now, it is different from death of a cell due to injury or something like that. Now, in case of cell death due to injury, we call it as necrosis. But in case of a cell deciding to die by itself, okay, that process is known as apoptosis. Okay. Now, mitochondria has a role in apoptosis. That is considered to be one of the most important functions right now regarding mitochondria. Now, how does mitochondria regulate apoptosis? Let me just come to that because that is something I think you'll be interested to know. Now, the mitochondrial outer membrane that I have just shown, okay, they contain a set of anti-apoptotic proteins, meaning those proteins that basically resist apoptosis. Okay, so this outer membrane of the mitochondria, they contain this anti-apoptotic proteins that protect the cell from apoptosis. Now, these anti-apoptotic proteins are neutralized via proteolytic digestion by a set of pro-apoptotic proteins, okay, resulting in the release of an enzyme known as cytochrome C in the cells that are destined for apoptosis. Now, cytochrome C, okay, that is released by the mitochondria, induces the formation of an apoptosome that ultimately leads to activation of the caspase pathway, okay, that brings about apoptosis. Okay, so any cell that wants to basically that decides to die is not allowed to die. It basically this process is finally regulated by the mitochondria. So that is why I say like mitochondria has a big role in the regulation of apoptosis. Okay, next slide, please. Next cell organelle that I want to talk about right now is the endoplasmic reticulum. Endoplasmic reticulum, they are structurally, network of membranes in the form of flattened sacs. As you can see in this diagram, they are shown as flattened sacs, or they may sometimes be flattened tubules also. Okay. Now they are connected basically to the nuclear membrane on one side, and on the other side, of course, they are extended throughout the cytoplasm. It's interesting to note that the endoplasmic reticulum constitutes more than half of the membranous surfaces within the cytoplasm of most of the cells. Okay, next slide, please. The endoplasmic reticulum, as you all know, must be knowing already, is of two types: the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is basically responsible for protein synthesis because they are associated with another cell organelle known as the ribosomes. So protein synthesis actually happens in the ribosomes, and of course, since the rough endoplasmic reticulum is associated with the ribosomes, they are also going to be involved with protein synthesis. Now, outer surface of the rough endoplasmic reticulum, okay, they are studded with ribosomes, and they are responsible for the production of all these proteins, the secretory proteins, membrane proteins, meaning proteins that I just talked about in the cell membrane. Okay, they are all produced by the rough endoplasmic reticulum, and also many organellar proteins. On the other hand, smooth endoplasmic reticulum, they are extensions of the rough endoplasmic reticulum. Okay, and they basically form a network of membrane tubules, but however, they lack the ribosomes that we see in rough endoplasmic reticulum. Now, this smooth endoplasmic reticulum, they contain unique enzymes that make it functionally more diverse than the rough endoplasmic reticulum, and they are responsible for the synthesis of fatty acids and steroids such as estrogens and testosterone. Okay. Now, proteins synthesized in the ribosomes. I'll talk a little bit about the rough endoplasmic reticulum. Proteins synthesized in the ribosomes, they enter first within the rough endoplasmic reticulum for processing and sorting. What the rough endoplasmic reticulum does is it basically takes in the protein synthesized by the ribosomes, and once inside, these proteins are processed and sorted out within the rough endoplasmic reticulum. Then finally, they are incorporated into membranes of organelles, or inserted into a plasma membrane, or secreted via exocytosis as the requirement may be. Okay. Now, next slide, please. Oh, okay. Uh, Coming to the Golgi complex, Golgi complex is another cell organelle, cytoplasmic cell organelle. Structurally, they are flattened membranous sacs. Okay, flattened membranous sacs with bulging edges. You can see in this diagram, they are flattened membranous sacs with the bulging edges. Okay, often giving the shape of a cup. Okay, these particular cytoplasmic cell organelle, they are more extensive in cells that secrete proteins. Now they have got basically two phases. One is known as the convex phase or the entry phase, and the other one is known as the concave phase or the exit phase. Now the convex or entry phase, it faces the rough endoplasmic reticulum, and the concave ones, that is the exit phase, they face the plasma membrane. Their orientation is such within the cell. Now, between the entry phase and exit phase, you have got these structures, okay, this cup-like structures of the Golgi complex. They are known as the medial cisternae. Now, enzymes, many enzymes are present, okay, within the medial cisternae that are going to be helpful in functioning in the functioning of the Golgi complex. We'll come to the functioning of the Golgi complex in the next slide. Now, the mechanism by which Golgi complex works is the entry phase basically receives, and modifies those proteins produced by the ribosomes that are attached to the rough endoplasmic reticulum. And once inside the Golgi complex, these proteins, okay, they go into the medial cisternae. In the medial cisternae, uh, carbohydrates are added to those proteins to form glycoproteins, or sometimes lipids are also added, may be added to those proteins to form lipoproteins. So within the Golgi complex, this addition of carbohydrates or lipids into those proteins that enter the Golgi complex happens to form these different types of lipoproteins and glycoproteins. Now, the exit phase, okay, modifies the molecules further. Okay, whatever modifications that has been done in the medial cisternae, okay, once those substances reach the exit phase of the Golgi complex, okay, further modification occur, sorting of those things will happen, and finally packaging also occurs in the exit phase for the transport to their respective destinations. Okay. Okay, next slide, please. Coming to the steps of protein processing in the Golgi complex, this is a picture showing that. Okay, I've taken it from this book. What we see in this diagram is you have this endoplasmic reticulum. These are the rough endoplasmic reticulum, of course, and then you have this ribosomes attached here. The ribosomes have synthesized this particular protein. Now, this protein basically comes out through the transport vesicle formed by the budding off of this portion of the endoplasmic reticulum. Okay. Now, this transport vesicles finally reaches the entry phase of the Golgi complex. Okay, entry phase, also known as the cis phase. Now, once inside, this protein undergoes a lot of modification, okay, within the medial cisternae, because medial cisternae has got all the enzymes required for the processing. So the processing happens there. Finally, the processed part comes out into the exit phase of this cisternae, and finally, this sorted out, packaged, and modified proteins. They are either secreted as secretory vesicles or membrane vesicles or transport vesicles as the need may be. And finally, they are sent out to their designated respective destinations. Okay. All right. Next slide, please. Now that was the function of the Golgi complex. Now coming to the fourth, I would say, cytoplasmic cell organelle, that is the lysosomes. Now structurally, lysosomes are membrane enclosed vesicles, and they are basically formed from the Golgi complex, budding off from the Golgi complex. Now these vesicles, known as lysosomes, they contain a number of enzymes. Okay, so far about 60 powerful enzymes, digestive enzymes, and hydrolytic enzymes have been uh, found to be present in the within the lysosome. Now these enzymes, of course, they need an acidic environment to basically work optimally, and the pH required for its maximum functionality is pH of 5. And to maintain this acidic environment, what within the lysosomal membrane actively pumps in hydrogen ions from the cytoplasm, so that the uh, pH inside the lysosome is maintained at 5 for these enzymes to work at their optimum. Okay, so this is a picture of the lysosome. As you can see, uh, it is basically a membrane enclosed structure, and inside you have got this different enzymes. Okay, there are about 60 enzymes, hydrolytic as well as digestive enzymes. Okay, present within the uh, this organelle called lysosome. Now, there's a term called autolysis. This is something I wanted to highlight. That's why it's there. Now, autolysis is nothing but a process by which an entire cell is digested by the lysosomes present in that cell. So it's amazing to think about one cell organelle that is capable of, digesting the whole cell itself. So that happens. That ability is there in the lysosome, and the process is known as autolysis. Okay. Coming to the next slide. Okay. Functions of the lysosomes. See, the first function, as you can see from the slide, membrane transporters, okay, move the final products of digestion, like the glucose, the fatty acids, and amino acids into the cytosol. Now, uh, uh, this first function, okay, is concerned with the digestion of this final products. This substrates, digestion of these substrates. Okay, and finally they are released into the cytosol. Second function of the lysosome is related to removing of the worn out cell structures within the cell. Okay, so these lysosomal enzymes, they are basically able to recycle those worn out cell structures. Okay, recycle in the sense like they are basically digested, and then like they are the byproducts are being released again, and those released byproducts are further recycled to form new structures. So basically lysosomes help in recycling of worn out cell structures. Now coming to the third function of lysosomes. Lysosomes, they are involved in cellular differentiation. When the cellular differentiation, meaning like uh, the cell getting differentiated from one form to another, okay, that is cellular differentiation. Cell growth, okay, that is also controlled by the lysosomes. Tissue remodeling, tissues get remodeled, okay, in that lysosomes have a role, and adaptation to adverse environments, and also cell defense, okay, via the process known as autophagy. Now regarding autophagy, I would like to tell you something. It is a very interesting subject, because it is something for which Nobel Prize was awarded to physiologist, okay, on his research on autophagy in 2017. So you should look up about autophagy. Okay, coming to the next slide, please. Now, this is a diagram showing the steps of autophagy. So Autophagy meaning not digesting the whole cell. It is about digesting the cellular contents. Okay, uh, worn out cells and things like that, cell components and things like that. Okay, first of all, in the first step, what happens is when autophagy has to start, something a structure known as a phagophore is formed. Okay, in the cytoplasm. Now this phagophore is an incomplete, enclosed chamber. Now it will basically move and then enclose those structures to be digested. Like for example, in this case, I'm seeing mitochondria and other cellular organelles also that needs to be digested. Okay, so the phagophore basically encloses those structures that need to be digested to form what is known as the autophagosome. Now, once autophagosome is formed, it will basically go and bind with the lysosome. Okay. Now, once it binds with the lysosome, uh, what is formed is known as the autolysosome. Now, once the autolysosome is formed, whatever hydrolytic enzymes the lysosome is having will be poured inside the autolysosome, and finally, there will be lysis of all these products that were taken inside. Okay, and they are basically released later on, okay, with the help of an enzyme known as permease. So this is how autophagy happens, and I think from this diagram you must be clear how what are the steps through which autophagy occurs. Okay, go to the next slide, please. Then coming to the peroxisomes, it's another cytoplasmic cell organelle. They are double membrane, again 0.5 micrometer diameter cell organelle, also known as microbodies. Okay, they are believed to be formed by replication of self-replication. They can form by self-replication, or they can be formed by budding off from the smooth endoplasmic reticulum, rather than, of course, from the Golgi apparatus. That is being believed. Okay, they contain enzymes known as oxidases, capable of combining oxygen with hydrogen ions to form hydrogen peroxide. As you know, hydrogen peroxide is a very potent, substance to digest things. So hydrogen peroxide, of course, once formed, it will associate with another enzyme known as catalase, which is another oxidase enzyme that basically will be in a position to oxidize most of the toxic substances. Now, one good example of this process is detoxification of alcohol, okay, into acetaldehyde by the hepatic peroxisomes. Okay. Now, major function of peroxisome is to catabolize long chain fatty acids. This is the most important function, but the second function will be basically to detoxify toxic substances. Okay. Now, coming to the next slide. So proteosomes, these are also another cytoplasmic cell organelles, very tiny. Okay, they are they are shaped structurally. They are barrel shaped, consisting of basically four stacked rings of proteins. They have a central core. Okay, now they function continuously to destroy, unneeded or damaged or faulty proteins that the cells do not need anymore. Their role is very important in the sense that they do not allow accumulation of all this unneeded damage or faulty proteins, accumulation of which may lead to disease conditions later on. Okay. Now these tiny cell organelles, they are basically present both in the cytosol as well as in the nucleus. This is something peculiar about this cell organelle that you should remember. Okay, they are present both in the cytosol as well as in the nucleus. Since the word itself, proteosomes, okay, so they have the ability to basically digest things. So they contain a myriad of proteases, that is enzymes that are able to cut proteins into smaller peptides. So that's how they are able to basically destroy all this unneeded damage or faulty proteins. Okay. Now, one application of, medical physiology application of proteasome is faulty proteasomes. They result in the accumulation of clumps of misfolded proteins. Okay, in the brain cells of people with Parkinson's disease, which is a very deadly disease, okay? And of course, Alzheimer disease. They come in old age most of the time, but these diseases, they are associated with faulty proteosomes. Now, once the proteosomes are not working properly, okay, there will be accumulation of clumps of misfolded proteins in the brain cells. Finally, these clumps of misfolded proteins, they are going to produce these disease conditions in humans. Okay, next slide, please. Okay, coming to the centrosomes, these are membraneless organelle. Please remember, this is the only membraneless organelle. Okay, this is something you have to remember. Rest of the other organelles, they were membrane bound, but centrosomes, they are membraneless organelle, and they consist of pair of barrel shaped centrioles, and of course a pericentriolar material will be there that will constitute the centrosomes. Now, I said they are made up of pair of centrioles. So that means two centrioles will be basically making a centrosome. So one centriole, okay, when you look at the wall of one centriole, it is going to be formed by longitudinally arranged, equally spaced 9 triplets of microtubules. So these are the microtubules you can see. Okay, you can think of this microtubules set of 3 microtubules. Okay, making one bundle, and then such set of 3 microtubule bundles. Okay, if you take 9 such bundles and arrange the way it is arranged here, okay, that will basically form the centrosomes. And one centriole, and then 2 such centrioles basically will be making your centrosomes. Okay, now that means centrosomes are made up of finally microtubules. Now these centrosomes, they are also known as mm, microtubule organizing system. Okay, so they organize microtubules basically, and microtubules grow basically out of the gamma tubulin in the pericentriolar material. Now, what is the role of microtubules? I'll be talking about microtubules when I talk about the cytoskeleton. So just leave it for the time being. Okay. Now, they duplicate. Okay, and pairs move apart to the poles of the mitotic spindle during cell division. That means centrosomes, when it is time for cell division to take place, they will duplicate. Okay, and then the pairs, they will move apart from each other to the poles of the mitotic spindle, finally leading to the uh, separation of the cell, okay, into two daughter cells. Now, centrosome abnormalities, okay, if there is any abnormalities in the centrosome, they can cause cancer, reproductive disorder, brain disease, and of course, ciliopathies. Ciliopathies are nothing but inability of the cilia, the cells that contain cilia, okay, that they are not able to function properly. So those conditions where the cilia of the cells having cilia, they are not able to function properly. Their cilia are not able to function. That is known as ciliopathies. Okay, because those cilia are basically related to, they are organized basically by, they are formed by these microtubules. Okay, going to the next slide, please. Now coming to the cytoskeleton. Okay, these were all the previous slides that I was talking about. They were all cytoplasmic cell organelles. Okay, now we'll come to the cytoskeleton part, which is of course going to be a component of the cytoplasm. Now, what is it? Cytoskeleton, they are nothing but system of fibers that maintain cell structure and allows cell shape to change and move. So this cytoskeleton is basically the fibers, okay, that basically maintain the cell structure. The cell needs to be in proper structure for it to function properly. So that function is going to be provided by the cytoskeleton. And of course, when needed, the cell can move with the help of these cytoskeletal structures. Now, cytoskeleton basically of a cell basically is made up of 3 components. Okay, the microtubules, microfilaments, and the intermediate filaments. Okay, three components: microtubules, intermediate filaments, and microfilaments. Now, out of these three, microtubules and microfilaments, they provide complex lattice work around the cell organelles, so that the cell organelles are also in the shape that they are supposed to be. Okay, come to the next slide, please. So just as I have just mentioned, the cytoskeleton elements, we have got three. They are the microtubules, intermediate filaments, and the microfilaments. Now, microtubules, since they are tubules, okay, the inside will be hollow, isn't it? So, and of course, if it is a filament, it will be all solid. So please remember that. Okay, intermediate filaments and microfilaments, they are solid structures, whereas microtubules, they have a hollow interior. Okay, so this is a microtubule. Okay, and this one is an intermediate filament, and then microfilaments. And I don't know why it is given like this. Now, microfilaments are made up of actin. Intermediate filaments, they are made up of several proteins. Okay, microtubules, they are made up of tubulins. Let's go to the next slide, please. Now, coming to microtubules, they are long, hollow structures. I just said they are tubules, so they are hollow structures with 5 nanometer walls surrounding a cavity which is having a diameter of 15 nanometers. So that means the diameter is more than the thickness of the thickness. They are basically made up of two globular protein subunits known as the alpha and beta tubulins. And sometimes they have a third subunit known as the gamma tubulins. Now, gamma tubulins, they were basically related to the centrosomes, as you must be remembering. Now, these tubules, microtubules, they interact with what is known as the GTP, guanosine triphosphate, to facilitate their formation. So, for the formation of the tubules, okay, interaction of microtubules, the tubules need to interact with guanosine triphosphate. Okay, and growth of the microtubules, you should remember they are temperature sensitive. So, when it is very temperature is high, microtubule growth uh, will be affected, or when it is low, again it will be affected. So microtubule assembly, you see, is prevented by colchicine and vinblastine. This colchicine and vinblastine, they are used as anti-cancer drugs. Okay, now as you know, microtubules are going to be associated with cell division. Okay, uh, these drugs, they target the microtubules so that the cells cannot divide. So they are used as anti-cancer drugs. Okay, utilizing the uh, physiological importance of microtubule, knowledge of physiological importance of microtubules. Okay, let's go to the next slide. Coming to the next cytoskeletal element, that is the intermediate filament. Now it is about 8 to 14 nanometers in diameter, and as I had mentioned earlier, they are made up of various subunits. Okay, not just one thing, but various subunits, and some of them connect to the nuclear membrane, to the cell membrane. Okay, and they basically function by to scaffold. Okay, scaffold that basically helps in resistance against external pressure towards the cell. So they basically maintain the cell shape and structure by their scaffolding effect. Now, proteins that make up the intermediate filaments, they are cell type specific. This is something you have to remember in medical physiology, because these different intermediate filaments subunits, okay, since they are cell type specific, this knowledge is being used to make these different intermediate filaments as tumor markers. So in different cancers, okay, one particular subunit, if it is found, okay, then we know that the cancer is from that particular cell type. Okay, so that is the significance of intermediate filaments in medical physiology. Please remember, now they are used as these subunits of the intermediate filaments. They are used as tumor markers. Okay. All right. Please let's go to the next slide. Coming to the third element of the cytoskeleton, that is the microfilaments. Name itself says it's filament, so it is going to be a long solid structure made up of actin fibers having a diameter of 4 to 6 nanometers. Okay, now these actin fibers, okay, they can be of two types: filamentous actin and globular actin. Now filamentous actin, they are found in intact microfilaments. Intact, okay, intact means when it is completely formed. Now the G actin, okay, or globular actin, they are present in the unpolymerized protein actin subunits. So they are still not yet formed completely, as you can see from this structure itself, isn't it? Now, F actin fibers, that is filamentous actin fibers, they attach to the cytoskeleton, okay, and interact with membrane-bound proteins. So the proteins that are bound to the cell membrane, okay, they interact. The F actin fibers, they interact with those proteins, okay, and they are most abundantly found in the lamellipodia. That basically helps the cell to crawl along surfaces. Sometimes the cells need to crawl along surfaces. For example, when hello, um,
Yes, Dr.
there's a lot of noise.
Sharma, you have,
Oh,
yeah.
is it past the time or what, ma'am? No, I'm just about to finish, ma'am. So I'll just go through. So this affectin, they are abundant in lamellipodia that help cells crawl along surfaces, meaning like there are certain cells, for example, the macrophages, they need to crawl in the tissues to function as defense mechanism. So in those cells, this affectin will be abundant. And some molecular motors that I'll be talking about now, they use microfilaments as tracks. Come to the next slide, please. These are the functions of the cytoskeletal elements: anchoring of the cell organelles and prevention of cell collapse, uh, helping in the amoeboid movement of cells, the spindle apparatus formation. Okay, then microtubules that are an element of the cytoskeleton. They form central parts of the cilia and flagella and contribute to structure and movements of the cell. They also form an amazing complex railway system inside a cell on which large organelles, smaller organelles, and certain large molecules, they are able to travel from one place to another, depending on the need of the cell. So these are basically the functions of the cytoskeletal elements. Coming to the next slide, please. Now this is what I was talking about, the molecular motors. Okay, these molecular motors, they are basically the vehicles through which proteins, organelles, and other cell parts, okay, they move from one part of the cell to the other. So they are able to move. They are basically the cargo carriers. They are vehicles for these substances to move, or organelles to move from one part of the cell to the other. They are basically 100 to 500 kilodalton ATPases, meaning they are having ATPase activity. Now, as you can see in the diagram, uh, you see this molecular motors, the cargo that is to be carried, attaches to the cargo binding domain. Now, the cargo binding domain is, you can see here, this is the cargo binding domain of the molecular motor. So the cargo binds there, and then the head, which is this is the head. It has got two heads as shown here, okay, in this picture. So the heads, they basically are bound to either the microtubules or the actin polymers. Okay. Now, since the head has got the ATPase activity, okay, ATP in the cytoplasm is being utilized. Okay, basically the ATPases in the head, they will break down the ATP and convert them to ATP, releasing an high energy phosphate group, getting the energy to basically move the cargo along the cytoskeleton, getting the energy by ATP hydrolysis. Okay. So most important molecular motors that are of significance to our cells, human cells, are the kinesin, dynein, and myosin. Okay, they are the three superfamilies of molecular motors. Let's go to the next slide, please. Coming to the cell junctions. Okay, in this diagram, we have got the three different types of cell junctions. One by one, I'll try to explain. Cell junctions, first of all, let us try to understand what is a cell junction. Cell junction is nothing but connection between the neighboring cells. So a connection between the neighboring cells or the contact between the cell and the extracellular matrix. Okay, so it can be either connection between the neighboring cells or contact between the cell and the extracellular matrix. Now, there are three types of cell junctions that I have shown here in the picture. The first one is a tight junction. Now, tight junction is basically a type of cell junction where you can see,
Thank you, Professor Sharma, for such an outstanding, thank you, Professor Sharma, for giving such an outstanding presentation on cellular biology. Thank you once again. And we have reached our next is Professor Yngkhom Govindaraj Singh, who is an distinguished professor in the Department of Physiology at the Regional Institute of Medical Science, India, and he completed his MBBS RMC Imphal in December 1988 and earned his MD in Physiology from RIMS under Manipur University in February 2006. Dr. Singh has been registered with the Assam Council of Medical Registration since January 1991 and the Manipur Medical Council since October 2015. His academic career at RIMS began in July 2006 as a demonstrator, and he progressively held positions as assistant professor, associate professor, and professor, accumulating over 27 years of teaching experience. In addition to his teaching roles, Dr. Singh has actively participated in medical education training programs, including workshops on educational science and curriculum implementation. He has held several key responsibilities at RIMS, such as being the officer in charge of the Central Animal House, convener and member of the Institutional Animal Ethics Committee, nodal officer for Kayakalp Swachh Action Plan, and a member of the School Board of Medical Sciences at Manipur University. He has also served as the public grievance officer and has been involved in teaching and examining several kind of students. His scholarly contributions include 3 international, 5 national, and 5 international publications, highlighting his commitment to medical education and research. Sir, you such an amazing personality, and I would like to request you to please start your session, sir. Thank you.
Good evening, everyone. Thank you, Dr. Sarita, for the very elaborate introduction of me. Am I audible? And can you see the screen? Hello?
Yes, you are audible, and uh, we
Can you
can
see the
hear
screen?
you. No, not yet.
Okay.
Can you present,
Mhm.
please? Yeah.
Is it working?
If there's a glitch, I think Dr. Moitru can help you out. He'll be presenting for you.
Can you see my screen?
Not yet. Yeah. Yeah.
Great, sir.
Good. Yeah. Great.
Can you see?
Yes. Yes. Can you go to presentation
Yes, sir.
mode?
Your screen is good.
You can see, no?
Yes. Yes.
Okay. Good evening, everyone. This evening, I'm presenting on a topic, the cell membrane and the membrane transport, which I will include some parts of the structural components of the cell membrane also. The cell membrane is a tri-layer as seen by two polar hat of phospholipids and an inner lighter of SRJ, which when I project it, it will be seen like this, which is a fluid mosaic model, which is most popularly described by Gert L. Nicholson and S. J. Singers in 1972. This membrane is also called a plasma membrane. It is a very thin, about 70 to 100 angstrom in thickness, double layered of lipids with interspersed proteins and carbohydrates in a mosaic form. This mosaic form, it envelopes the cytosol and the cell organelles into a compact structural unit of life. The phospholipids are arranged, positioned side by side of each other in each leaflet, buckled by a number of proteins and cholesterols and some portions of carbohydrates. The functions of the cell membrane is mainly to insulate the interior of the cells, that is, the protection of the cell from the damaging effect of the environment and other multitudes of molecules entering and disturbing the cell. It provides mechanical support by connecting with the neighboring cells with the help of many connective tissues, which are often known as the cell junction, and with the other connective tissue around the cells. Another function of the Cell membrane, yes, it has a role in keeping the homeostasis between the inside of the, that is the intracellular compartment of the cell, and the outside extracellular compartment of the cells by involving many biochemical actions and activities status for the optimal functioning. It also involves the maintenance of the fluid and volume homeostasis by exchange of water for its optimal function. A cell cannot survive alone. It can also have, must have a way of communication with the other cells so that the communication will keep alive the functioning of the cells, which will be stressed later on, and which is mainly related with the immune system. Now, there is a lot of chemical reaction also going on in the cell membrane, in the outer membrane surface also, and both in the inner membrane surface of the cell membrane. And it is for the maintenance of all the events which are occurring around the cell membrane. Then there is a cell adaptation, because the environment of the cell is sometimes very hostile and sometimes very altered, and it has to adapt to remain surviving and to adjust itself to the ever-changing environment, and it has to maintain the morphology. For example, there is a nice example of, say, the RBC, the cell sporulation of RBC. What is sporulation of RBC is, sometimes when the RBCs are traveling in the bloodstream, there can be sometimes damage of the membrane by hitting on the cell wall or with the others, and because of the stream flow. And because of this damage, the cell membrane, it has got its spontaneous ability, its own capacity to mend up or to repair the damage in the cell membrane. Now let's come to the components of the cell membrane. The cell membrane is composed of a protein which is 49% of the total mass in dry weight. And it has got a phospholipid with 33% and a cholesterol of 11%, together forming the 44% of lipids. And the carbohydrate is a mere 7%. Of course, the molecular number of the phospholipids are much, much higher than that of the protein in number of moles. Now let's have a brief account of the membrane lipids. The membrane lipids are divided into three types, which is known as the phospholipids, glycolipids, and the cholesterols. These membrane lipids are amphipathic molecules. Amphipathic means the molecules have got both a hydrophilic, that is, which can be attractive to the water, and a hydrophobic, that is, which can be repelling away from the water molecules. These are called amphipathic molecules. These phospholipids have a polar hat. The polar hat has a polar group with a certain phospholipid compounds with a phosphate section and a glycerol section, which connect between the polar hat and a portion which is known as the hydrophobic portion, which is known as a fatty acid chain. This is a long fatty acid chain, which I'll be describing later on. Now, the two leaflets with this phospholipids are contiguous to each other in each leaflet are arranged spread out in such a manner that there is a mixture of different types of these phospholipids all around outside in the cell outer leaflet also, in the inner leaflet also. But there is a difference between the nature of the phospholipids in the outer and the inner in this molecular structure, in the types. Now they are arranged in such a manner that the long acyl chain, it's a the fatty acid, which is the hydrophobic. It is formed with around 14 to 24 carbon molecules chain and variable according to different types of cell membranes in different types of cells having different types of cell membranes. And these are kept in position to next each other by various hydrophobic hydrogen bonding, van der Waals forces, and many other hydrophobic this mechanism. This some of the phospholipids are the phosphatidylcholine, the then there is the sphingomyelin, then there is the these are phosphatidylcholine and sphingomyelin and this thing and some other phosphatidylethanolamin like this. They are mainly they are neutral charge carrier. They are neutral mainly. But some phospholipids like the phosphatidylserine and phosphatidylinositol, they carry a negative charge. The properties of the phosphatidylserine is very important when there is a system in which whenever there is a change in the nature of the membrane while going to transport or going to accept some molecules and others, there is a phenomenon known as the flip-flop. That is, the phosphatidylserine, which are mainly found in the inner membrane, can be changed over to the outer when some activities concerning with some molecules and attachments occurs. This which is known as a flip-flop. Flip-flop. And there is another property of the cholesterol, which is another which is another lipid. They are found impacted in between groups of the phospholipid compounds in such a manner that when they are impacted like this, they erase the phospholipids in groups and thus inhibiting from unnecessary migration of the phospholipids laterally. This is a very important. The phospholipids have also got a very, very important role in changing their influence on the other phospholipids, the cholesterol, and also on the proteins which will be found amongst them. The diacyl phospholipids, which are seen here, it's an example of diacyl phospholipids with two long acyl chain, and these are the major components of the membrane lipids. The sphingomyelin, for example, is abundant in cells like neurons, where they may be almost composing 80% of the phospholipids in the neuron. Whereas in some, it can be another type like the diphospholipid, diacyl phospholipids. They are found plenty in the mitochondrial membrane, for example. Then the cholesterols have also another functions in protecting the structural reorganization in the phospholipids. Some of the phospholipids can be also a source for the one making event which is known as the signal transduction, in which they supply some of the breakdown and then some phosphate energy sources for them. This is another property of phospholipid. Anyway, let's come to about the sugar portion. The sugars, that is the carbohydrates, are found mainly on the outer membrane leaflet, and it is very much rare in the inner leaflet membrane. In the outer leaflet membrane, the glucose molecules can be various with galactose, mannose, acetylgalactosamine, glucuronic acid, and lots of them. Simply glucose molecules can be also there, and they're in long chains. They protrude from the outer membrane mainly, and those which are associated with the proteins are very often known as, like this one, is known as the glycoprotein, and those which are associated with the lipids are known as the glycolipids. And examples can be found in the blood groups where the fructose is also involved, and which determines the ABO grouping system like this. They are all glycoproteins. And there is another one which is known as the salic acid attachment of salic acid. Is a long uh, carbohydrate chain which are found associated with another integral type of protein which is known as the glycophorin. And this salic acid are very much responsible for keeping they're found in the cell membrane of the RBC, and they're very important for keeping the cells apart, that is repelling from each other so that they don't come into contact and uh, alter in the cell functioning of the RBCs. Now let's come to the membrane proteins, which are of usually are of three types. The integral protein, which spans throughout the outer membrane leaflet and the inner membrane leaflet, are actually they're structurally a trans amino peptide chain in many turns, which are known as the motif or domains. And many of them are the proteins are just the long polypeptide chains. They're arranged in a coiling structure known as this one like this as known as the alpha helix coil. And in some cases, the proteins, the amino peptide chains, are arranged in a ribbon-like structure, which is known as the beta pleated. And the importance of these proteins in arrangement of this one is because the side chain of the amino peptides of various amino acid residues here will be arranging such that the negative, positive, positive, positive like this in a various manners, so that the proteins will be either in an open state, closed state, or in a narrower one, and like this in different configuration according to the state of the cell membrane, this one, status of the cell membrane activity, which I'll be coming again, how it is done. Then there is the peripheral membrane protein, which are very much close to the inner membrane, and these are represent many at times, many enzymes, and many other proteins which are involved in signal transduction, like the um, G protein coupled receptors like this, which is a transmembrane protein, and which are actively taking part in the signal transduction, that is transferring signals coming from outside the cell to inside, giving a message to the nucleus for their activities. And there are, and apart from this integral membrane proteins and the peripheral membrane proteins, we have got the an extrinsic type of proteins, which are mainly located in the outer membrane protein. These are also known as the globular proteins. And these globular proteins, many of the globular proteins are the one which are going to associate with the carbohydrate chain. And the one which are found peripherally in the inner membrane is also, those proteins are also called as the intrinsic protein also. So in order to, now let's come to the membrane transport after a brief explanation of these different structures. Now we have come to the membrane transport. And in order to understand the fundamentals of the membrane transport, as well as the mechanisms for the development of electrical resting membrane potential, there is a voltage potential difference in the cell membrane between the outside and the inside of the membrane. So let's try to understand it. Let's try to recall some basic processes of what is diffusion and membrane permeability, that is allowing substances and solids and molecules to transport across the membrane. The molecules of gases, liquids, and dissolved solids in motion are continuously at a random motion, which is known as the Brownian moment. As you can see, the arrows directed in all directions. Now, when it is movement, the movement of this each and every molecule how far it moves, how strongly it moves, in what direction it moves, it depends on the some factors like the thermal effect, that is the heat or temperature, and the types of molecules which are around it. But whatever it is, the molecules or the solutes which are suspended in a solution, for example, when they're moving and swinging, they the displacement, they may move this side, come back again, go this side, come back again. So the final displacement is always at zero. It looks as if it is not moving when it is in a gaseous state. The velocities of these individuals, molecules, are very highly variable because of that variable and also the kinetic energies which they give up. Now, the momentum and the energies are always preserved, even though they appear to be in a very highly moving motor in motion, and it's not losing any momentum. It is preserved all the time. Now, in such a condition, if the molecules are partitioned into components with unequal numbers and molecules. Now, in this condition, what happens is that because there of the accumulation of the kinetic energy in one area where the solids or the molecules are concentrated in relation to the other side of the compartment where there are relatively lesser number of molecules, the kinetic motion is less. So it imparts a strong influence of movement because of the energy evolved and the momentum it gives out to tendency to move from this one to this. If the membrane separating is permeable to this solid, then it will start moving towards the lesser concentration side. So when the molecules tends to move from the higher concentration to the lower concentration, it will continue until it becomes equalized in both sides, and which is known as the molecular diffusional equilibrium. And the process in which the movement of the molecules from the higher concentration to the lower concentration, because of this dent of difference in the concentration gradient, is known as the diffusion. And here, the driving force is the concentration gradient. The flow continues till the distribution equalizes, as I say. And as we can see, a very good example when there is an oxygen which are found plenty in the alveoli, when it goes to the pulmonary capillaries through the lung capillary membrane, which is permeable to the oxygen. It's a very good example where the alveolar oxygen concentration and the pulmonary capillary oxygen, they equalizes during inspiration. Now, the diffusion through the membrane, because of this, the only limit, if there is going to be any problem, then the only limit is going to be the membrane. So we used to say that diffusion through the membrane is the rate limiting state when the membrane intervenes. Because of the membrane characters, concentration gradient of the solids are, and accounting the thermal state, the flux of the solids follow. That means the membrane, if there is a change in the membrane, then naturally when the molecules are flowing from a concentrated area to a lesser concentrated area will be a little bit altered. And it's because it follows a law, which is known as the fixed law of diffusion, in which when the molecules are let alone there is lots of collisions of the molecules and lots of this direction which altered the momentum instantaneously. Of course, it comes back to its original all the time. But this becomes more prominent when a membrane always intervenes between these different concentration gradients. So there is a difference in the alteration in a flow of the concentration, flow of the molecules from the higher concentration to the lower concentration. And this flow is termed as flux. That is the J symbolized by J. This flux depends on, first, as usual, whether the one thing is the concentration gradient across higher concentration on one side and lower concentration. But because of the intervening membrane, there is the area of the membrane through which the molecules will be penetrating through it. And then we have come to the thickness of the membrane. As I was telling you, because of the intervening membrane, the thickness matters. And the higher the thickness, it is inversely, the flux is inversely to the thickness. That is, the higher the thickness, the flux is lesser. And the thinner the thickness, the flux is higher. And this is known as the thickness of partition, we can say. And there is a D, constant diffusion coefficient of the solid. The diffusion coefficient of the solid is nothing but for every solution, solute, and for every molecules. And it's a standard thing in which for a given concentration gradient, that is for a given means with a non-specific unit concentration gradient, in per unit square centimeter per every second, and with the temperature and pressure fixed, there is a flux value which is very specific for that molecule. This is known as a coefficient, diffusion coefficient. So if you want to determine the flux of any solid, then all we have to do is we have to know the concentration of gradient across the area of membrane, thickness of membrane, and then multiply by the diffusion constant of that substance. So if there is a concentration of the inner and out, if molecules are coming from this diffusing out here, the difference will be, if this is higher, this will be minus the outside the concentration divided by the differential of thickness, that is the small thickness, with the temperature and the pressure constant. So we can see that there is so many factors can affect the diffusion of a membrane. So, but whatever it is, with a certain fixed membrane thickness and others, the fixed law of diffusion applies. Now, when the membrane, the diffusion coefficient is very, very fast, and the molecular size is very small, and in the membrane with a fixed membrane, there is a favorable for the smaller molecule to pass through to diffuse through the membrane in a faster than another complicated molecule, which is larger and more heavier to pass through it. That is, the diffusion coefficient is least. So because of this, the diffusion coefficient of those neutral things, okay, which is non-ionic and others, even though they don't produce anything, then they will take it longer time to pass through the membrane. And they are most of the lipid soluble, since in the bilayer phospholipid, we know that the polar have got solubility. Those which are soluble to it, mainly the lipid things, can pass through easily. But what about the ions? The ions are, though they are very small in size, there is one thing called a water cloud. The water cloud are things which are surrounding the ions and which forms a bulky thing. And when the water cloud comes and trying to penetrate through the diffusion, there is the hydrophobic region in the central which will repel them away. So the process things for these ions, they find it difficult to diffuse through it. So they require special other accessory things for them to pass through. So one thing is, in short, we can say the diffusion coefficient of a substance in free solution is affected upon molecular size, shape, and charge status. So we come to one condition which is known as the selective permeability. This permeability refers to the ability of a cell membrane to regulate the passage of different substances into and out of cell. So the transport of a substance through the membrane requires another thing called permeability apart from its property of diffusion. This characteristics allows the membrane to permit certain molecules or ions. As you can see from this figure, you can see that some of the ions, the green figures, can pass through easily. These are selected because they are favorable to the membrane, they are lipid soluble, or they are not restricted by the hydrophobic, and they can pass through easily. They may be very small molecular size and with a high concentration with a driving force. But there perhaps a problem with red figures with representing some molecules which the inherent properties may be either they may be very big size, or they may be having ionic charges, or they may not be a lipid soluble like this. So when they come, because when there is a, if we think that there is a high concentration, the membrane is there, they cannot pass through. Rather, they are repelling. This is called reflection of it. And so the permeability is which allows a selective molecules to be allowed, and certain because of unfavorable things are repelled back. This is known as the selective permeability. So in the selective permeability, the things, what regulates the selective permeability, this is called, because of the first thing is the lipid solubility of the substance in the lipid, which is not, when it's not there, the membrane bilayer phospholipid itself can be a this one block to the transport. Now another thing is that for other molecules, the membrane, cell membrane has a property, has this quality of having these proteins which I've explained, which will facilitate, which will help in the transport of other substances and other unfavorable things, which is known as the transport proteins. The more these transport proteins are expressed in the cell membrane, this means those which were not allowed can be this one. But these are very specific. So they will not allow anything. They will be specific for some certain types of other substances and molecules. The size of the particle. For the size of the particle, sometimes if the particle is required to be transported to the other side of the cell membrane, then somehow it has to be taken up by the cell membrane and will be transported. Then there is a charge of the particle which will be repelled away because of the presence of the hydrophobic. Then there is a term which is known as, which I mentioned here in the transport protein, the specificity. With every molecule, this transport system has to be a specific mechanism. So this one is for the permeability. There arise the permeability coefficient. The permeability coefficient is nothing but, suppose let's take this as a cell membrane. When this is a cell membrane, there is a outer cell, outside the inner of the in the cytosolic portion, and the membrane here, phospholipid. There is a gradient here. There is a problem. And on the other side also of the cell membrane, there is the outer, the external and inside of the cell. So there are two barriers here, okay? And when a particle which is moving from a location inside, and it's going to transport to the C location outside, when it's going to, there is going to be some hesitation or some reluctance of this for a free movement of it. And this is because of the gradient between some barrier effect between this outside of this membrane in the inner side and inside of the cell membrane. And if you take the difference in this, the reluctance, you can say the resistance, you can say in this side and in this side, if you take the ratio between the two, that is known as the partition coefficient, because this is partitioning. And it is represented by beta. Now, if this beta, if you take into account, because this membrane has become very important as a part of the permeability, and if you take it, multiply it by the diffusion coefficient, and it is divided by the distance through which it's going to travel through, then this is known as the permeability coefficient. And this permeability coefficient is very, very determinant of different types of this protein molecules transport from one side to the other side. And for the
Sorry to
membrane,
interrupt, Dr.
and
Govindaraj. You
yes, yes.
have just another 5 minutes left to wind up.
Okay, actually, I
Please,
think I've gone into only half an hour. Anyway,
please, because we have
okay,
only 2 hours time,
okay.
right? Yeah,
Okay, I'll quicken it up. Okay,
yeah.
okay, I'll quicken it up. Okay, so the selective permeability is very important for many various purposes. It's for the fundamental property of biological membrane, essential for processes such as the nutrient uptake, for the waste removal, that is the metabolite waste removal, for regulating the tonicity of cells in the environment. Then there is a potential difference across the membrane, that is the voltage difference across the membrane, to be built up by the involvement. Now, these functions of the membrane proteins are also required for the lateral mobilization, and it's also, apart from its most important, transport of substances across the membrane. Now, the functions of the membrane protein are can be classified under ion channels, pumps, facilitators, substance transport, receptors, energy transducer, enzyme, immune, and the configuration of the proteins are going to change according to the type of transport. Ion channels, for example, the ions are going to be transported from one side to the other side. Okay, and the pumps are against the concentration gradient. The ion channels may be down the concentration gradient, where the ion pumps is reverse of that against it with the use of an energy ATP molecule. And there can be other in which some proteins can help as a facilitator, that is in helping in the diffusion in the which is known as facilitated diffusion. And others are more involved in immune reactions and opening and closing of many things. Okay, the role of transport protein, as we know, involves various transport mechanisms and ions and substance across and through the membrane. Now, the transport is divided into passive transport, that is driving force of transport, down the concentration gradient, and an active transport by use of energy. Now, this mechanism of transport protein is the closing and the opening of a channel which is located inside, deep inside the membrane protein. And this is because of the presence of it, and it can be by various mechanisms in which a ligand is a substance which have got a receptor, a loci where there is affinity for the ligand to come and attach to it, which will change the configuration into an open state and allow ions to go inside. Another is when the hormone, for example, when it touches, the inside of cell can change its phosphorylation. That is, with the use of a ATP molecule, it can phosphorylate, energize, and it can open up. And then once it opens up and do its function, it goes back to its dephosphorylation. And another is voltage gating. It's an interesting one in which if there is a change in the membrane potential of the cell membrane, the configuration changes and this open up. So, and another is there when there is a mechanical traction, there is a shearing force acting on the cell membrane, there can be a close to an open state. So there are various ways in which the membrane protein can open up, close, open, according to the type of stimulation. Now coming to the transport of passive transport, we have got a simple diffusion, a facilitated diffusion, and osmosis. The simple diffusion is nothing when the molecules are spread across with the membrane which is permeable to molecule, and when suddenly this membrane is exposed, because of the concentration gradient, it starts diffusing through it. When this diffusion, then until they come into a equilibrium, this diffusional equilibrium. This is known as a simple diffusion, which I have explained before. And because of the different ionic charges, molecule size and others, they have got different conductivity. That is the diffusion coefficient with the potassium more than sodium, even though they have the greater atomic weight because of the lesser watery cloud, okay. Now we have got Another one in which a facilitated diffusion, in which the proteins are found help. This is the channel in which the proteins are going to be diffusing through it, this one, but with the help of another protein, okay, which is known as the protein transporter. The from a position of E1 configuration, when the particular substance comes close to it and attached to it, then suddenly it changes into an E2 position in which the substance is thrown inside. And it is done, the advantage is that this transport protein will increase the fastness of it and in a well more better regulated manner. An example is the GLUT in a glucotransporter, which is found in the intestinal epithelium. And this such a saturation point can be reached following a Michaelis-Menten reaction with the substrate and the product balancing. And another is with the channel mediated ion transport, which are found in many of the cells, which is known as a sodium channel leak, potassium channel leak, and these are purely ion channels, which goes on without any transport protein, but their concentration present wholly and according to from the outer to inside, because the outside the cell is a 145 millimole per liter in outside of the shell extracellularly, and inside is hardly 15 to 20 millimole per liter. So the sodium goes down in these leak channels, for example, this one, and this is going to be corrected by another protein with an active transport mechanism. Then we have got the osmosis. The osmosis is a low tonicity solution, okay, with a high tonicity solution. Means when you're comparing between the two with a membrane partition between two, there is more water molecule and lesser solid. This I call a low tonicity and high tonicity. But the water molecule is more in the low tonicity, so it tries to diffuse to it, and it diffuses through special water channels and aquaporins and special water channels in between the membrane, which is very slow, of course. And since deep inside is a hydrophobic, it will repel break. Otherwise, uh, in order to avoid that problem, they pass through the water channel. And these water molecules move until and unless the pressure or the tone in the both the sides are equalized. This is known osmosis, and the pressure which is developed when it is coming to the transport is known as osmotic pressure. Now we come to the active transport. The active transport is for transport of many large substances or small molecular sized particles and ions, but transport in bulk by a vesicular active transport process by cytosis. So we have got different active transport. One type is the endocytosis, pinocytosis, and phagocytosis, which is known as the vesicular active transport. And there is another one which is called exocytosis according to the direction of the cell movement of the transport. There is a transcytosis. This is a phagocytosis in which the bacteria, when it is coming, it's a foreign organism, there is pseudopods which are extended out, take it inside, digest it with the help of lysosome, and then it come back to a normal. This is a phagocytosis and eating, cell eating. And there are other clathrin-mediated endocytosis, molecules helping in the this one expressed in the inner side of the membrane, which will favor the movement of a particle inside it. Then there is when it goes inside from the extracellular fluid to the intracellular component. And there is a pinocytosis, which is known as a cell drinking when water is required in bulk, and which is very often occurring in the endothelial surface of the intestine. Then we have got the exocytosis, in which from the proteins and others enzymes which are formed from the Golgi endoplasmic reticulum, this one pathway, it is being transported to the cell membrane, and it is being released into the outside, which is known as exocytosis, that is from inside to outside. And we have got a transcytosis in which the particles are being taken in a vesicular active manner with the involvement of energies, and it comes down and brought to the other side of the cell, which is in the basal side, and to be transferred further into the capillary. This is in many cases in the absorption of nutrients, that calcium, glucose, and amino acids, and in the intestine also this transcytosis occurs. Now this So active transport, we have two types, the primary and secondary transport. And the use of energy, as I said that in the active transport, there is a against a concentration gradient with the use of energy. So a primary and secondary transport. This is a energy is used in the form of ATP and uh, in in other phosphate related to, containing energy, either directly or indirectly. This is the active transport, and there is an example of calcium ATPase which are found in the sarcoplasmic reticulum, for example, in the muscle, in which this is lower concentration, higher concentration, and against this. And this is with the use of an ATP converted to ADP with the inorganic phosphate carrying energy, changing the configuration of the transporter here. This is known as the CA, calcium ATPase. Now, there is a primary active transport which is known as sodium potassium ATPase pump. These are also known as pump, in which the more sodium, the inside outside of the extracellular is more in sodium, the inside is less. And this is opposite in less potassium outside and more. Now there is a leaking of saved ions also. Now what happened is that in case of the sodium potassium primary active phase, now when inside, because of a disturbance of the leaking of the sodium ion inside it, there is a disturbance in the sodium concentration. This disturbance is sensed by the sodium potassium ATPase, and three molecules of the sodium ions are abruptly attached to it. They have affinity whenever there is a change, since it's a very sensitive to the change in the sodium ion. This causes up to potassium ion, which was in an attached mode here, to be released. And this is known as, in a form which is known as an E1 configuration. And this, the moment the potassium is released when the sodium is, almost simultaneously, the sodium is thrown back up this um, outside, and there is a conformational change will cause the it will cause a phosphorylation in which one ATP molecule will be used for the ATP for these activities. The potassium, which is outside, which is very less, will be attached because of this energized. The potassium is less outside. More, it is around 150 millimole per liter in the inside. Now it is attached there, this. And this causes the sodium to be released and the potassium to be thrown inside because this one. And this form is known as the E2 and converting to E1, in which the the potassium, which is corrected the for the normal molecular concentration. And this is the one secondary active transport. And in the secondary active transport, we can see the sodium or potassium ATPase is going to indirectly help the movement of the ion channels, is going to build up an energy which is going to help in the transport of the sodium inside. And this movement of the sodium transport will help in the movement of a glucose molecules along with it in the same direction, and this is called SGLT2. This is found in intestinal. This is down the concentration gradient. Now we have come the co-transport. When the two molecules are moving in the same direction, we call it as an electroneutral symport, sodium chloride in the same direction. And for the electrolytic symport, with the use of an ionic change, we have got amino acid with the sodium ionic concentration. And where the ionic concentrations are not required at all, there are also movement of the electroneutral antiport also when they are in opposite direction. And there is a tertiary active symport, which is an electrolytic. And thank you for the present hearing. And these are the references which I have gone through. Thank you.
Thank you so much, sir, for an enlightening presentation. Your expertise has enriched the minds of students and enthusiasts alike, and surpassing the boundaries of the cell membrane and membrane transport. It's such an amazing presentation, and thank you so much. Now, we eagerly welcome Professor Praveen Kumar, sir, to bring this session to a remarkable close.
Yeah, good evening, everyone. Hope I'm audible to everyone. Am I audible, ma'am? Hello.
Yes, you're audible. Yeah, you are
You're audible,
audible.
sir.
Yeah, great. Once again, good evening, everyone. Before going for the vote of thanks, there is an announcement about upcoming lecture series, which is June lecture series, which is going to be held on 22nd June 2024 between 7 p.m. to 9 p.m. The topic is that immunology, and we are going to have our guest speakers as Dr. Kaushik Bharti, UNESCO. We'll be talking on the topic immunology and overview, and Dr. Sumi Mukhopadhyay, Calcutta School of Tropical Medicine. He'll be talking on the disorders of immune system. As we come to the conclusion of this enlightening guest lecture on cell physiology, it is my privilege to deliver the vote of thanks on behalf of the Education Committee, the Physiological Society of India. First and foremost, I extend our heartfelt gratitude to our distinguished guest speakers, Dr. Govindaraj Yngkhom and Dr. L. Amarnath Sharma, for sharing your valuable insights and expertise with us today, sir. Uh, Currently, I know that Dr. Al Amarnath Sharma is having the electricity issue, because of which, towards almost he's about to complete the presentation, which was abruptly stopped because of electricity issue, which we were facing in Arunachal Pradesh since morning. So, hope all of you excuse for this. And sir, Dr. Ramana Sharma and Dr. Govindra sir, your knowledge, and we are honored to have you had here. And a sincere thanks to our organizing team for your meticulous planning and coordination. We're also grateful to our participants for your active engagement and enthusiasm. And in conclusion, thank you all once again for your invaluable contributions and participation. Thank you. Have a great time.